not waste any time. Hi, everybody. My name is Sarah. You might know me from the previous editions of the MentaLABS webinar series. I'd like to extend a very warm welcome on behalf of myself and everyone at MentaLab to what I think is now the 5th edition of our MentaLABS webinar series, which is quite exciting for us. This is a monthly venture for us, and we're really enjoying it so far. And today we are very lucky to be joined by yet another wonderful speaker who we are in connection with. I'd like to introduce you all to Ms. Rochelle Aubrey, who is from KU Leuven, and or more specifically, NeuroTech Leuven. And she has a very exciting presentation to be sharing with us today. So just to give you a brief idea of the agenda for today, I will not bore you with any extraneous details, but I do want to introduce us to you, as well as the entire purpose of this MentaLABS webinar series. Followed by my not so tedious details, we will have the presentation by Rochelle, which is titled Neurotechnology in Action, from Brain-Computer Interfaces to Early Biomarkers of Parkinson's Disease. As you can hear, that's quite a mouthful, and I think you should sit tight because we have lots of information, of course, to share today. There's lots going on in this presentation. A variety of different topics will be shared, and we hope you enjoy it. Following that, of course, we will have a discussion and Q&A session, so please do keep your questions and curiosities all the way till the end, and you will have time to ask Rochelle all the questions that are in your mind. So just to briefly tell you a little bit about us, who are we at MentaLab? So MentaLab is a neurotechnology company based here in Munich, Germany. And what we do is we manufacture and produce these really nifty little EEG devices called the MentaLab Explore Pros. Our system is, of course, a naturally very high performance system with a variety of different sampling rates depending on the channel count that you're looking for. We have a lot of cool features with the device. So not only does it measure physiological data, but it also measures orientation data with an inbuilt IMU. There's, of course, lots of different technical details to share, but I cannot bore you with them now. But the main concept is that we are a mobile device. So we have a very small and lightweight device that weighs approximately 27 grams. This is our 8-channel system. It has a very high battery life, and of course we are optimized for mobile settings, meaning you can actually stream the data via Bluetooth BLE. But you also do have the option to do it wired in case you want to have a more standardized lab setup. We are also very proud of our very open source nature and approach to EEG research. So we have a desktop and mobile application, as well as a Python API, which is completely accessible to the researchers who are using our product. And there are no subscription fees or license fees hidden here whatsoever. And we hope this helps you out as well. So what is the purpose of our company? Of course, we are desiring to be innovators in the biosignal acquisition space. But outside of the standard manufacturing and development of our device, we also want to support research and development efforts in our community. So we support a variety of different researchers and institutions across the globe, ranging from the States all the way to the corners of Asia. And as you can see at the bottom, we have lots of different partner companies based here in Germany, in the US, in Asia, in Africa, many different locations. So you can probably find us somewhere, wherever you're calling from. And of course, what we're here for today is to foster a scientific exchange with our MentaLABS webinar series. So we are hosting this webinar to sort of spotlight the current research that is happening in the space and to stimulate a collaboration, as well as explore future directions in the EEG and EXG space. So without further ado, I would like to introduce to you today's speaker, Ms. Rochelle Aubrey. She is a PhD researcher and neuroengineer at NeuroTech Leuven, who specializes in a variety of different domains, as you can see from this very long description here at the bottom of the page. I do not want to take the spotlight away from Aubrey. I'm sure Rochelle, she will, of course, introduce herself as well and show you what she's working on. But just so you know, she is a very impressive individual with lots of cool projects to share. And so I think that's all from me for now. I'm going to pass over to Rochelle, and she can tell you a little bit about what she's been working on.
Thank you very much, Sarah, for all the introduction and explanation of MentaLABS. It's always so nice to see all the advances the MentaLAB has achieved. Indeed, I'm gonna share my presentation. Um, Perfect. Um, is it good? Is everybody able to see it?
Yep,
Perfect.
that looks good. Full screen.
So thank you. Yeah, as Sarah already mentioned, I'm a researcher at KU Leuven. I'm currently general supervisor of NeuroTech Leuven. We work with different type of brain signals from EEG. We are also starting to venture towards fNIRS. I'm more privately in my projects. I work with the neuronal, more specifically motor neuron signals for modeling of and understanding of ALS disease for drug screening purposes and personalized medicine. But today I will talk to you more about what the projects within NeuroTech Leuven that we have. So I've chosen mainly three of them, but we have much more projects going on in the organization, which we will be able to also tell you about if you're interested. But a bit first of like this organization was created because obviously there is a very big need to target neurotechnology, to target neural related disorders, since there is an increasing amount of people suffering a variety of different disorders. And we see a lot of potential for all kind of neurotechnology types of applications and that can be used to improve the not only the diagnosis, but also the patient's lifestyle. So we also saw that there was a very big need in education, since at least in KU Leuven and in a lot of the universities around Europe, we see that there is a lot of it that is taught in different courses, but it's not very clear for a young student and sometimes for even those more mature ones, what are the specific information that we need in order to leverage neurotechnology itself. So we thought that it was important to offer more of this framework, this accessible entry point towards the specialization around the different topics that neurotechnology offers. So a few years back, then I guess already 5 years ago, and this is the current team that we have. Every year we have a different team. Some of them remain, some of them graduate and go to a job in the industry or a PhD. But the main goal that we have at our organization is that everybody that comes to the organization creates a community that provide and foster between each other and amongst external people knowledge and passion about neurotechnology. And we've based this organization in three main pillars in which we can provide education, we can provide projects, and we can provide connection. So these three main pillars, at least for education, because we have a structure where a lot of the PhD students throughout KU Leuven or other universities are involved, as well as our people in the industry, they are able to provide hands-on learning to students, workshops, hackathons, throughout the whole projects, guiding them every step of the way so they can more efficiently work together as a team. We also offer some connection in the sense that we organize events ourselves to bring people together. We also go do company visits or lab visits where we get to talk to the engineers or the researchers at the different institutions we attend to, and to get to know what is the state of the art there and how we or students can help with the activities that we're doing. And finally, and the most important thing for us are the projects. That is what I'm going to talk to you about. So we have three main branches of projects. One of them is brain-computer interfaces. We usually gather more bachelor students here, but we also have had most of them master students in the past working on it, all guided by PhD students. Then we have our industry-oriented project and our research-oriented project. So the industry one is for people that are interested into going to have a job afterwards into different companies in the biotech sector. Then they can already come to NeuroTech in their studies and having a good idea of what is it to do there, or what is it to create a startup or start your own venture there. So you can have this opportunity with the organization. And then we have the research projects that are more like research oriented, of course, or like a taste of what would be to have your own PhD project or later on in the career. So all these projects again are guided by PhD or persons in the industry and proposed sometimes by professors or companies or have born within NeuroTech Leuven. So I'm going to present you three of them. I will go with in the first one a bit more in detail, and the other the two other ones, I will go more on an overall level, but you can always ask questions about the three projects. So one of the first projects that we have here is what we call the brain browser. That's what we named it. This is a beautiful team that worked in this project throughout a whole year. They look very happy here, but it was stressful times, I would say, for them from time to time. But what they achieved was a very interesting application that can be leveraged for patients with locked-in syndrome, ALS, or partial to full paralysis. The idea is that having a complication like that really isolates you from the real world, and you kind of lose connection with people that you care about, with people that you know, or you feel the frustration that you cannot really do much. So the idea is to improve this by offering the possibility to at least browse on the web, and this particularly on Instagram, to have more of this interaction with people. So this project is based on SSBP signals, so steady-state visual evoked potentials. That's why we see here the flickering lights at different frequencies with different colors, because we are also sensitive to color here. You can check all of these fully on our GitHub, which you can see the QR code here in the left bottom part. If you want to browse it, you want to even just start working with it or even improve it, feel free to do so. Everything is fully available. So this is mainly what this screen is mainly what the patient or the person using the platform would see. And in order to do that, I will share with you the top-down overview. So basically, we have a person with the headsets on their brain. Then the EEG data goes to a pre-processing parts. Then this goes sent via WebSockets to your web browser, so you can browse it through any computer. Then you receive the stimuli, which is this, at the different frequencies, which indicates one of the actions that you want to perform. Here you can like the post, you can browse up and down, and from this EEG signal, we will extract what kind of which of these inputs the person was looking at. So I will go more in detail into this. Because we're a steady-state visual evoked potential that are neural responses to visual stimulation that translate the same frequency band of that the person is directly looking at, which is the frequency of the liking the post, the going up or down the post. This by posing the electrodes in the visual cortex in the backside of our heads, we are able to detect these SSBEPs. This signal is then pre-processed in order to, of course, take out some environmental noise, the electrode natural noise, or some kind of potential glitches or blinking, or any ongoing other brain processes that can happen. Now, in our project, and if you browse our GitHub, you will see that we went to several pre-processing steps and several classification steps, but I will show you only one of them, one that I think is super interesting. In order to classify, we have to first pre-process. And for that, we, of course, do an electrode selection, which is, as I mentioned, in the visual cortex. We filter our data. And because of the methodology that we're going to use, which is Riemannian geometry, we're going to apply a bandpass filter for each of our a bandpass filter to filter out the noise, and a filter bank for each of the frequencies we are interested in. So here we use 8, 10, and 12 hertz. which is one of each button that you saw, the going up or down, is one of each of these frequencies. And then we epoch in order to have this data in meaningful defined time points. From this information, we will create some covariance matrices. This is very interesting because it allows us to see this the information from all the electrodes that we have here. Once we have our covariance matrices, we are able to create a Riemannian manifold. So before going too much into the manifolds, Riemannian geometry is it's a branch of differential geometry that allows us to specify data in a complex manifold, which you can see here. It's a complex manifold. The very interesting of manifolds is that it allows us to work with higher dimensional data and as if were a lower dimensional data. The potential of manifolds in brain data is that brain data is very complex and is not two-dimensional, but it has a kind of geometry, three-dimensional, multidimensional geometry innate to the complexity of the signals. You can use multidimensional manifolds to represent the activity of your neural population and have a very good estimate of your of the state of your signals by creating these covariance matrices in your Riemannian manifold. So after we do this pre-processing, we do a bit of a training. The training will estimate the covariances by the Lidoid-Wolff-Schinkel estimator on the extended signal that we mentioned before. The extended signal is the one that we created through a filter bank in order to obtain the data from your three signal buttons. Once we do that, we estimate the centroids using a medium distance to mean classification model. It's just a classification that you can use via Riemannian. And actually, Riemannian geometry, it's already it's already been implemented in different SSBEP methods, and you can use already open libraries available out there, open source. We use this estimation for the centroids because it's very simple, it's very robust to noise, and it's very efficiently computational. So we saw a great possibility there to use this metric for our purpose. So the centroids are then estimated according to a Riemannian metric, meaning the distance from each of these centroids. In normal machine learning or other methodologies, you use traditional Euclidean distances. But here we're using the Riemannian distances because we are considering that we are in a multidimensional space rather than in a two-dimensional space. And once we have this, we can do a prediction. So it takes a bit of learning, and it uses the fitted model to calculate this distance between our centroids. So I hope this is I'm able to somehow explain this very complex subject to you, but of course, feel free to go through our GitHub or ask me any questions if something of this wasn't clear. But following up on our pipeline, we already have established that we have a data acquisition or visual cortex of SSBEP signals. We have already established that we do a pre-processing that adapts to the different classification methods that we implement. Then we have different classification methods, in this case, the Riemannian-based classification method. Then we want to send this information to our web browser. So the web browser knows what kind of signal the person is looking at and able to act accordingly. So the WebSockets will not only connect, will serve as a bridge, and will send messages from processing to extension forward and upward in order to be able to actually perform the action that the person was intending. So it's a very straightforward closed loop methodology that runs real time on our platform. And of course, it's still this is a project that could be still improved. So feel free to check up on the GitHub link if you want to know more about it. Then I want to go a bit further to something that we have on Parkinson's disease. And for this, I want to present our other project in vibrotactile brain stimulation. This is the team that is mainly working with this project. We are collaborating with F2Hill. F2Hill is a organization founded by two engineers that are also suffering for Parkinson's disease because they saw that there is a need to have more open source ways to treat and diagnose the disease. So the main problem in Parkinson's is that you have a lot of rigid muscles, you have the tremor, you have the slowed movements, and most or at least large part of the disease patients receive a kind of medication. But there are also other methods that are not necessarily medicated because medication not always functions very well for patients or not always is sometimes, at least depending on the country that you are, it can be a tricky decision to make. Do you want a medication based that will work for a certain kind of time if it works, or maybe a deep brain stimulation or alternative stimulation technique that also might work depending on the extent of the development of the disease. So then we have deep brain stimulation, but of course it's invasive, it's expensive, and it's strict to patient-specific assessment. So there was a lot of very interesting research done there in that the possibility of using vibrotactile brain stimulation as a non-invasive, affordable, and accessible technology as a therapeutic for Parkinson's disease. So the only problem that it comes with vibrotactile brain stimulation is that it is not necessarily effective for every patient, and it varies depending on the patient itself. But it has been showing very interesting results in counteractive desynchronization that is traditional of Parkinson's and many other diseases. I can also mention ALS and has a strong synchronization neuronal problems that can be stopped and unlearned via this alteration of pathologic connectivity. So, but in order to do that, we have to see which are the frequencies that actually help and understand the effect of this vibrotactile in modulating brain activity. Are these frequencies that work better? Are frequencies that work less? Is a specific time points that have to be targeted, or maybe just some patients are just not necessarily suitable for this kind of therapeutic compared to others. But it's very important to highlight that this is an interesting approach because it is non-aggressive and it will not cause any type of impairment to the patient themselves. But it could ease this because of the connectivity that it has with the brain. So as mentioned before, there are different habituation, cognitive states, motor interaction, and mechanical factors that induce, that change the potential effectiveness of Parkinson's itself. So in the project, the goal, what we already have achieved is that the engineers at F2Hill have already generated a vibrotactile stimulation device. It is as well all open source for things that one can buy and integrate ourselves. This is also with the means that people suffering the disease, not necessarily with the economical means, can still access to this potential type of therapeutics. The idea here is that we find a way to achieve the desynchronization of the disease states of our brain or neuronal communication systems in order to reduce the symptoms of Parkinson's disease. So how do we do this? We apply the vibrotactile stimulation with different conditions on healthy individuals, of course, and then we go towards the patients suffering Parkinson's disease in order to find how this is affecting our brain signals. And this is where we leverage the EEG signal as well. Is this stimulation causing any kind of improvement visible in EEG or not? Or is it causing more distress than anything? So, which usually the stimulation should be in a range that it would not cause distress, but we want to optimize the good effects that it may cause. Here I can show you a video of our team working with this device. So as you can see here, this is the vibrotactile stimulation that they are using by now. They are coupling the EEG headsets, and they're performing the stimulation at the same time that they're doing the measurement and obtaining some relevant data that they can investigate to see if they could see any benefits in the changes of the synchronization of our brain signals. So our project is ongoing. This is a project that is for us is lasting 3 to 4 years, given the extent of the things that we have to take in consideration, and of course all the ethical approvals that we have to go through, etc., etc. So of course we are taking a research roadmap for it. The first one, of course, is evaluate the effect of vibrotactile brain stimulation using EEG signal. Find an EEG-based Parkinson's biomarker, and optimize the BBS for long-lasting effect on the patient themselves. So yeah, we will happily share more updates on that. You can always look at our websites if you're interested more, and then we will publish more results once we have them. But with this, I conclude this project, and I would like to continue to the third and last project that we have within our organization. This is more of a research project, and this is in collaboration with Neuroelectronics for fNIRS, which is now part of the BIB here in Leuven. And this is our beautiful team as well working on it. Let's go. So I've already talked to you about Parkinson's disease before, and yeah, we know it's a strong disease that affects a lot of people throughout the world, that has very few options when it comes to treatment. It has very early degeneration of brain cells, with the problem that symptoms occur very late. Once the degeneration is usually irreversible, of course, and then it has the latent difficulty diagnosis that we already mentioned about, that we are looking to target. So our idea or goal with this project is to find as well, as in vibrotactile is more, okay, which biomarker we can use for EEG, with EEG, so we can actually see if there are benefits or not with this treatment potential. But with this project, it's like, okay, can we be able to diagnose it earlier so we can use techniques to treat it better throughout their lifespan, so the life expectancy can be longer, or the life quality of the people can be better. So we can see our previous project more of a treatment-like project, and this would be more of like diagnosis-based project, as well as kind of understanding or if whether it can be used or not, if there may be a better method, and if there is a better method, which one could it be? So it serves many purposes. As well, we have it's also a project that will last 4 or 5 years for us, if anything. Same reasons than before. It's a lengthy process. But we are focusing in olfactory dysfunction here because it is very highly associated with neurodegeneration. So clearly, as well as age, of course, but in neurodegeneration, we see a clear trend that a very high amount or percentage of of patients, we estimate that 70% of the patients suffer some kind of olfactory deficiency at some level of the disease. This coupled with other things as well that could be served as co-biomarkers, but I'm gonna focus on olfactory dysfunction because it's very interesting. So yeah, we've seen that also it's not necessarily in Parkinson's. We also see that Alzheimer's has the same problem. While we're focusing now in Parkinson's, this could be potentially in future trials extended to other neurodegenerative diseases. So there do exist a method currently in the market and in the doctor office to evaluate the loss of smell. And this is the sniffing sticks. It's the traditional thing that you would go if you go to the doctor and you say, "Doctor, I don't smell anymore." Okay, they will do this test. It's a sniffing stick test of 12 or 16 different aromas. You will have to qualify to determine, to basically you have different identifications. So either can you identify which odor is it? Can you identify the threshold, meaning do I smell this concentration? Can you identify between two of them? So these are a few of the metrics that they use to analyze. That's why you have to fill a few of these papers, and depending on the score you have, okay, you have a diagnosis that is between some range. So hyperosmia, normosmia, anosmia. The range, as you see, is quite high, and this is something very subjective, which is something we don't really like when doing diagnosis, because the subjectiveness of these tests can be that you change with time. You as a person change and are more sensitive or less sensitive to different aromas, or of course age has an important role here, which we lose this capability to smell, and other aspects in our life can change the way we perceive these aromas. So so the TDI score, which is the way eventually we get the score, it can vary and it not necessarily be an objective measure of the loss of smell throughout time. So do we think, is there another way in which we can use target this or approach it? And can we differentiate between Parkinson's or Alzheimer's olfactory loss with age-related olfactory loss, or can it be used for Parkinson's disease? So we are performing these projects in which we have an olfactometer. So this is a very nice machine that allows us to present different odors to patients in a very controlled manner. So we know the concentration, we know the moment, the exact moment in which we deliver this smell to the patient directly to their nose. And then we measure both EEG signals and fNIRS signals. And then we use these data from our anosmic or Parkinson's affected subjects to see whether or not we can identify any of these smell changes throughout time. And for this, we stimulate our patients. We know that in Parkinson's disease, at least, there is an amount of there is a differences between the others. We know that even though after they have lost most of their smell, they can still sometimes detect very pungent bad odors. So that is very interesting for us to test different kinds of pleasant or unpleasant odors to see there is a change there. Also, if we are very close to one smell or other, if we have an emotional or a memory banded connection to these others, that can also have a different intensity in how we perceive the odor. And of course, concentrations, because even two healthy individuals can have very different TDI scores without the need that this TDI score variation be related to any kind of disease. Just habituation, training, current state of the person can change this. So we're stimulating our subjects with this, measuring simultaneously with EEG and fNIRS. We like to use EEG here because it offers a very high temporal resolution on the olfactory event-related potentials. And we can extract all very interesting components that are known to olfactory stimulus. So the initial sensory processing of the other, the perceptual and conceptual sensory evaluation of the other, and the responsive cognitive or memory factors to the smell. So it gives us very interesting information that can correlate several of the variables that we know about other. We couple it with the fNIRS. With fNIRS allows us to have to obtain the changes in oxygenated and deoxygenated blood on our prefrontal, or orbital cortex. So this is a multimodal approach that will give us both the temporal and kind of a spatial kind of information that could be relevant for detecting smell. Given that none of these techniques is super perfect for detecting signals, given that we know that the olfactory bulb is quite deep in the brain and can be a bit of a challenging. That's why we're trying to complement these multimodal techniques to have a better understanding. So yeah, for this we use our MentaLABS headset and our fNIRS headsets, and of course we create this database for diagnosis. Now, what things are we looking there on both our EEG data? So we're seeing if that we can see some latency shifts, which we would expect that it's delayed in Parkinson's disease because they have a loss of this olfactory capability. We also probably expected to see some changes in beta oscillations maybe, or network efficiency given the what we know about Parkinson's. While with fNIRS, we're expecting to see whether we can observe some patterns of activation that vary between the orbitofrontal or the prefrontal cortex, or as well, just very interesting to see if we see clear changes in hemoglobin responses. Again, this is coupled with a very exploratory, very interesting, with hopefully a nice potential application further in the roadmap. I want to also show you here a bit of our video of our lovely students getting some signals. You can see here the nasal catheter. That's cannula, sorry, where he's receiving a controlled amount of a smell, a very pleasant in this time, but hopefully in the future some unpleasant as well, so we can see the clear differences. Yes, so this is our third project, and with this I want to, of course, thank all the team. I think there are pictures of more teams that I haven't added in this presentation, with whom all this has been able to happen. And of course, our dear sponsors, Leuven, Medtronic, MentaLABS, that always helps us with their EEG headsets. And please feel free to send me an email or anything if you ever want to collaborate or work in any of these projects together with us. Thank you very much, Melis and Sarah, for the invitation. Let me know if there are any questions.
Thank you so much, Rochelle, for this presentation. It was really cool to both see some technical aspects and also both the Parkinson projects and how they approach the topic in different domains, like tactile, olfactory was really cool. So we thank you for this amazing presentation. And then now we could move into the Q&A session. And if there are any questions, we could take them via chat, or I think potentially there should be also a button to request raise hand. So if you would prefer to verbally ask them, please feel free to do so.
I think maybe I could start it off since there might be a few minutes of people typing their questions, if you don't mind. I have a very, I suppose, a bit of an anatomy question for one of the projects. So relating to the Parkinson's project, which I think was incredibly cool. Like Mela says, it's amazing to actually see our device being used in so many different types of domains and different applied settings. So thank you for showing all of that. Again, really, really cool. My question sort of stems from my background in more of a molecular neuroscience. I saw that in the Parkinson's project, you showed a diagram of the somatosensory pathway. It's totally fine if you don't want to dig into these kind of nitty-gritty details, but for me, I'm just curious to know whether the vibrotactile stimulation exerts this effect within this pathway, if you maybe have anything to add to that. So I'm interested in just the theory. So how did vibrotactile stimulation come to mind, and does it exert its effect in this pathway at a very early sensory processing stage, or is it maybe later on in the cortical networks? Totally fine if this is completely out of scope.
No, no, don't worry. It's perfectly fine. I think it's a very interesting question. Well, we know that stimulation happens very early on already in the fetus, in the development, and that any kind of stimulation is able to shape the way we are, and there is a very strong role on plasticity. When it comes to stimulation. Now, we know that the stimulation in different areas of the brain can promote this plasticity to rewire some of these pathways. And but of course, it is a very highly complex and very long set of connections happening in this one in a specific. So I don't know how much rewiring would there happen to be in this specific pathway. We do know that there is a clear connection between it. Um, and it has been reported that some patients respond very well to this vibrotactile stimulation, but it depends a lot on the kind of frequency that is given to them.
Mhm.
I know at a very more molecular level, the way stimulation works, at least I can say for ALS, is that when we apply some kind of stimulation, we can kind of liberate this tethering that is happening in between the membrane and the extracellular ambience in order to liberate the accumulation of unnecessary ions, because in a
Mhm.
lot of this is we have the misarrangement of ion channels within the membrane,
Yeah.
or they just don't work the way they should be. In the case of ALS, the axon detracts, and I think also in Parkinson's and Alzheimer's, you will have the same, that axons start failing, the ions start failing, the ion channels start failing, and an unnatural accumulation of ions lead to irradical and very synchronous set of excitation or activation of the neurons, creating a very heavily environment, very toxic, because it requires a lot of energy, a lot of neurotransmitters there, creating these toxic environments. So what vibrotactile stimulation, DVS, or some kinds of stimulations aim to achieve is that you kind of liberate this on normal accumulation of ions by kind of initiating this liberation, so it can go to a more rest state.
Hmm. Yeah, I think I know. meaning, yeah.
But I don't know if that somehow answered your question. I went a bit
No,
too
definitely.
far-fetched through
I,
it.
of course, with Parkinson's as well as ALS, it's a multi-layered problem. There's no one solution
Mm.
for anything. So your answer was incredibly sophisticated and definitely did answer what I was asking. So thank you. That's from me.
Thank you.
Are there any other questions in the chat, Melis?
I don't see one yet, but maybe I could ask one. So I come from a psychology background, so I'm not really familiar with the computational aspects. So this might be a well-known concept, but I was really interested and surprised at the complexity of the logic between the manifolds and the Riemannian manifold, etc. And as someone who is more on the psychological side of the field, I wanted to ask whether this approach of approaching brain data as something multidimensional and analyzing in such sense is relatively a new trend, or has it been an approach that has been used for a while?
Yeah, I think the paper that have the first use manifolds approach for brain data, for EEG data actually in monkeys in order to understand learning pathways, which the discovery was amazing. That they saw that data that lies on this manifold, the brain connections that lie within this brain manifold are more easily recruitable for us in order to learn compared to those that lie outside of this manifold. So this paper was, I think, one of the first one using this concept, and it was around 2014, 2015, I think. So maybe I think so, if I'm right. So it already has a few years there. And I think a few years ago, it's kind of being used a bit of in a buzzword, kind of become a popular thing. Uh, but I think it makes a lot of sense, although it's difficult to sometimes mathematically explore it fully, because we can make a lot of assumptions that are not necessarily true, or difficult to have such a high dimensional and kind of lower it from to lower dimensions where the data can be more easily analyzed can also take away a lot of the benefits. So it's a bit of like how much complexity is good to actually answer what we want to ask. But so yeah, that's the tricky part. But I would say there's been around and there's been super interesting research there in order to understand brain data with this. So yeah.
Really cool. Thank you so much. And we have a question, and we have a few questions in the chat. I could read them out if you would prefer. So the first one is coming from Matt, and it says, "Thanks a lot for the presentation. For the SSVEP, have you considered using the harmonics of the frequency of the interest too? In my understanding, the bandpass filters were only for the frequency itself and not the harmonics. Also, huge congrats on the sophisticated SSVEP approach." And a smiley face.
Thank you very much. Yes, actually, the harmonics is a magnificent further step. It's something that we wanted to work towards, but I think we implemented for our canonical CCA classification model because we implemented two. But yes, harmonics is definitely something to look at when analyzing this is the data that we had. Thank you.
Great. Thank you. Another question we have is, in your opinion, what are the biggest limitations to SSVEP-based BCIs at the moment? Data quality, ability to integrate SSVEP in natural environments, etc.
Yeah, indeed. I think noise is a big problem. It works very well in a very nice environment where there is no noise from phones, from people, from all of these. I mean, we have to be realistic about the how much can we actually measure from there, how deep into the visual cortex can we get, and how well our processing of the data can be. I think SSVP is still one of the good ones there because it offers kind of a very direct way to probe the brain. But still, yes, I think both that you mentioned are very strong still difficulties, kind of like obtaining reasonably good data. Uh, yes, but if we go to more deep brain electrodes, then we have the problem that encapsulation, and then okay, then well, always difficult, I would say.
Exactly.
Thank you for the question.
We will have one more question. And this one is from Sergio and is as a follow-up of Sarah's question, have the brain circuits affected with vibrotactile stimuli already identified? Have other fingers, in example, tested in this approach and its correlation with other vibrations that can be explored?
Sorry, can you repeat the beginning?
Yes.
Maybe I can also see. I don't know. Sorry. Uh, yes, sorry. You can repeat it. I don't know which one I should read.
Yes. Have the brain circuits affected with vibrotactile stimuli already identified? Have other
Okay.
fingers? Yeah.
Okay. Yeah, I think we are assuming, or we know that there is some kind of stimulation, direct somatosensory pathway that we can target there. That's but of course, there is still so much variability, and I think still we still need to find a very clear reproducible, like kind of extendable biomarker that we can actually achieve with EEG. So I would say there's still something that we're trying to forward, and we are trying all the fingers in our analysis in case there is extra sensitivity that we can target there. Um, but yeah, I would say it's still quite ongoing in that aspect.
Thank you. And we have one more question. This one's from Nitu. I hope I'm pronouncing it right. And this one is very interesting work. You're understanding olfactory sensation in a multimodal fashion. Are you planning to correlate both fNIRS and EEG data? Because both have a different sampling rate. Is it possible?
Yeah, that's one of the indeed the tricky parts that we have in the project. We nevertheless are trying to find some kind of other measurements that we can implement at the same time to kind of kind of like a good markers, of course, that we can kind of flag for us to understand the data. Um, we do expect that, of course, fNIRS will have a lag, and we don't necessarily know the lag that correlates to our olfactory signal. Um, uh, so yeah, working with the paralyzation would also require kind of very standardized other co co going metrics to implement there to see if we can kind of mark the beginning of both or some aspect like that, or kind of work with some kind of set grid instance delay kind of metrics or epoching to work with. But that is one of the challenges we are facing.
Thank you so much. For now, that appears to be the whole list of questions, and we still have a few minutes. So if anything comes up to mind, any questions come up, please feel free to share them. And otherwise, I guess we could wrap it up slowly.
Thank you for the questions.
So thank you, Rochelle, so much for joining us today. It was really cool to see all the variety of projects and how also our devices are utilized in them. And we are truly really insightful session, so we really enjoyed it, and we hope that attendees also enjoyed it. If any follow-up questions or anything else, please feel free to contact us through either one of the emails. And there appears to be something in the chat, so maybe we have another question. I'll quickly check that. Sergio, thanks for the presentation. And I guess that's about it. Sarah, would you like to add anything? Rochelle, would you like to add anything?
I think from my side, I'd just like to extend another thank you to Rochelle for everything. Really, it was awesome to just hear about everything going on at KU Leuven, and we wish you all the best with the rest of the projects. Of course, like you said, these are long timelines and lots of work to still be done. So please do keep us updated on all the happenings in Belgium. So yeah, thank you again. And I would just like to mention to all the attendees as well to just stay tuned for the next webinar series that will be coming at the end of June and early July. So just keep an eye out on our LinkedIn page and also on our newsletter for any further updates. So I think that's all from our side, and we can wrap it up today. And hopefully people are maybe at home by now or can go home for an early day, early. But thank you very much for joining, everybody. Thank you again, Rochelle. Thank you, Melis, for everything.
Thank
And
you.
we will see you next time.
Thank you. It's a pleasure to be here. Thank you very much for the invitation. Thanks all.